So I'm here with you with a very short session. One of our friends had asked a little small doubt about the modified Lac Operon model, how it is used in genetic engineering, that is the alpha complementation. So I'll quickly talk about that very briefly. Before that, before we go there, you should understand what exactly is there in the original Lac Operon. Okay, so this is the original Lac Operon in front of you. As you can very well see, it is having the multiple genes. You have the Lac Z gene, most important, then we have the Lac Y, the Lac A. These three make up the Lac main enzymes which will get transcribed, will participate in the metabolism of lactose. We have additional gene Lac I, which acts as the repressor for the whole of the Lac Operon system. This is the original structure of the Lac Operon. All of you know this. Now, in the genetic engineering, we use a modified Lac Operon where we introduce a not exactly a cut, we take out a small part of this Lac Z gene. So you can see here, we take out a small part of this Lac Z gene and we put this small part into a plasmid. Alright, we put this small part into a plasmid. Now when the regular transcription for this modified Lac Z gene takes place, it is missing a sequence of the nucleotides, and therefore it is missing a sequence of the amino acids in the transcribed protein. This is therefore the inactive beta galactosidase, because a small part of the functional domain is missing. The deleted part which is there in the plasmid, once it gets transcribed and translated, it creates a small fragment. We call it the Lac Z fragment. Okay, the Lac Z fragment interacts with the inactive beta galactosidase, and together they form the functional enzyme. This is the active beta galactosidase. Okay, and in this particular model, we provide the analyte which is X-gal. This is white in color, and if it is metabolized, means if the active beta galactosidase is assembled, in that case the X-gal will get converted into the metabolite which is blue in color. This is blue in color. Okay, so this is the alpha complementation. This is the phenomena of alpha complementation. We call it the alpha complementation because the alpha fragment, this is the Z fragment, which is the alpha fragment, complements the function of the original enzyme. This is the alpha complementation. Why we be using this complicated system? Because this Lac Z fragment which is here, it has a very simple restriction site. Restriction site can be easily opened up using the restriction enzymes. We open up this restriction site and use it to insert a target gene segment. When you want to do the genetic engineering, we use this Lac Z in the plasmid, introduce a targeted segment. If this insertion is successful, the product which is formed, that is the mutant alpha peptide, it is not able to support the action of inactive beta galactosidase, and it will not convert the white colony into the blue colony. Okay, so wherever the white colonies persist indicates a recombination. Wherever the blue colonies are transformed, blue colonies are being formed, that means the alpha complementation is still taking place, but we are interested in the recombinant colonies. So when the white colonies are achieved, this is the stage of transformation. The Lac Z fragment in the plasmid has been transformed. It has taken up the target gene that we have provided. So the base Lac Operon model, it is modified to create a sample, or you can say system, where the alpha complementation comes into play, and it is used in genetic engineering where a target gene segment is inserted into this Lac Z fragment in the plasmid to create a specific product. the phenomena will be transformation. So these are the two names you have to remember. Transformation, where we are looking at the successful integration of the target gene segment in the plasmid. Alpha complementation is a model which will work, and this whole process is called the blue white screen. Blue white screening is the classical method to identify the gene transformation when you're doing the genetic engineering. I hope this clarifies the very important topic of the modified Lac Operon and its use in the genetic engineering. If you have any more doubts, just ping me, drop a message in the chat, or you can drop a message directly to me on my groups in the Telegram. We are available on Biochemistry by Dr. Nilesh Chandra. You can reach us on Facebook. You can reach us on Telegram, or you can reach us on Instagram, where I'm present as Nilesh Biochem. Thank you very much. Keep learning. Bye bye.